Something 40 feet long is moving through the forests and river channels of what is now the American West, on two legs, unhurried, because nothing sharing that ground has any reason to make it hurry. Its skull alone stretches 5 feet from snout to crest, packed with teeth built less like blades and more like a batch of railroad spikes. When it closes its jaws, the bite lands with roughly 8,000 pounds of force, concentrated at the tooth tip into pressures of over 400,000 pounds per square inch, enough to punch straight through solid bone the way a modern predator punches through soft tissue. For roughly 2 million years, Tyrannosaurus rex sat at the absolute top of the food chain across this entire landscape. Not one apex predator among several, the single largest, most powerful land predator its ecosystem had ever produced, effectively unchallenged. Then, 66 million years ago, an asteroid ended it, along with every other non-avian dinosaur on Earth. What it left behind was bone and teeth, scattered across the American West, and in one specific almost impossible case, something that should never have survived at all. The obstacle that defines Tyrannosaurus rex research at the molecular level is the DNA itself. Ancient genetic material has a hard ceiling on preservation. Under the most favorable conditions, cold, dry, chemically stable, DNA survives approximately 1 to 1.5 million years before it degrades beyond any known recovery method. Tyrannosaurus rex went extinct 66 million years ago. That gap is not close. It is more than 40 times beyond the outer edge of what any known process could preserve. Whatever genetic information this animal carried is gone, permanently, in that form. Claims of dinosaur DNA recovered from amber-trapped insects, the premise behind an entire film franchise, were never independently replicated by the scientific community and are now considered contamination. But in 2005, a team led by paleontologist Mary Schweitzer found something else entirely inside a T-Rex femur, soft, flexible tissue that, by every rule paleontology had operated under for a century, should not have existed either. What Schweitzer found was not dinosaur DNA, and it never would be. It was a different, sturdier kind of biological evidence, structural tissue, and eventually fragments of a specific protein called collagen. Collagen is tougher and chemically more stable than DNA, built from a triple helix structure that resists breakdown in ways a fragile double strand of genetic code cannot, and under narrow, specific conditions, it can survive on a timescale DNA never could. That distinction is what makes the rest of this story possible. Inside a fossil preparation lab, the leg bone at the center of this story, a Tyrannosaurus rex the team nicknamed B-Rex, had been broken in the field, simply because the plaster jacket protecting it was too heavy for the helicopter to carry out in one piece. That break exposed a cross-section of bone recovered from the Hell Creek Formation in Montana and dated to roughly 68 million years old, that would otherwise have stayed sealed and unexamined. Schweitzer's team published their full findings in the journal Science in March of 2005. If you're finding this interesting, be sure to subscribe to understand more DNA from the past. The scientific community's reaction was not celebration. It was caution, bordering on disbelief. Schweitzer herself was careful not to overstate the finding, and other researchers pointed out that the team had not yet proven the material was actually dinosaur in origin, rather than some kind of contamination or bacterial growth that had colonized the bone long after burial. The skepticism was reasonable. Nothing in the accepted chemistry of fossilization predicted that this should be possible. Schweitzer's team answered with more evidence rather than argument. In 2007, working with mass spectrometry specialist John Assara at Beth Israel Deaconess Medical Center, they extracted actual protein fragments from the T-Rex material, 6 distinct peptides, adding up to just 89 amino acids in total, sequenced directly from collagen preserved inside the bone. Mass spectrometry works by breaking a protein into fragments and weighing them with enough precision to reconstruct the exact order of amino acids that made it up. When compared against a database of modern species, those fragments matched the collagen of modern chickens more closely than anything else tested. The following year, in 2008, a follow-up analysis led by Harvard researcher Chris Organ ran that same sequence data through formal phylogenetic software, comparing it against protein data from 21 living species, including elephants, alligators, and multiple bird species. The result placed T-Rex, using molecular evidence for the first time in history for any non-avian dinosaur, on the family tree directly alongside birds, closest to chickens and ostriches, and more distantly related to alligators. This matched exactly what skeletal anatomy had already suggested for decades. What it added was something skeletal anatomy alone could never provide: actual molecular confirmation at the level of protein sequence that birds are living dinosaurs. That level of publicity invited a serious challenge. In 2008, a separate research group published a paper proposing an entirely different explanation: that the structures Schweitzer's team had found were not dinosaur tissue at all, but bacterial biofilm, a slimy colony of modern microorganisms that had grown inside the porous, empty spaces of the bone long after burial and simply happened to mimic the shape of ancient blood vessels. For a period, this explanation gained real traction in the field, and for good reason. Biofilms are common in porous, buried bone, and distinguishing a colony of modern microorganisms from genuine ancient tissue under a microscope alone is genuinely difficult. It offered a mundane, testable alternative to a claim that otherwise sounded closer to science fiction than paleontology, and it could not be dismissed without direct evidence. To answer the biofilm challenge directly, Schweitzer's team then examined a second specimen entirely, an 80 million year old duck-billed dinosaur, excavated and rushed to the lab quickly enough to minimize any chance of modern contamination. The tissue recovered from that animal was even better preserved than the original T-Rex sample. Its morphology, its immunological reactions, and its amino acid sequences were all consistent with genuine animal tissue, not bacterial growth. Independent researchers, using an entirely separate technique called in situ mass spectrometry, confirmed the presence of hydroxylated proline in the tissue, an amino acid that serves as a specific signature of collagen, one that bacterial biofilm simply does not produce in that form. Two separate specimens, two separate research teams, two separate analytical techniques, all converging on the same conclusion. The biofilm explanation did not hold up. What remained unanswered for years afterward was not whether the tissue was real, but how it could possibly have survived. It is widely accepted among molecular biologists that protein molecules, even under favorable conditions, should not persist for longer than about 4 million years before breaking down past recognition. 68 million years exceeds that ceiling by a factor of well over 15. Standard chemistry offered no mechanism to explain the gap. And for years, the preservation itself remained just as mysterious as the original discovery, an open question with no accepted answer sitting underneath an already controversial finding. The resolution, when it came in 2013, turned out to involve the one component of blood that never really goes away: iron. Subscribe to not miss the next one. Schweitzer's team identified iron particles intimately bound to the preserved soft tissue in two separate dinosaur specimens, detected using electron microscopy and X-ray techniques capable of mapping individual atoms. Iron is abundant in blood, carried inside hemoglobin, and the researchers proposed that when an animal dies, iron released from breaking down hemoglobin drives a chemical process called the Fenton reaction, generating free radicals that cross-link surrounding proteins into a tightly bonded, highly stable structure. In practice, this is remarkably similar to the way a lab preserves tissue with a fixative like formaldehyde, except in this case, the animal's own blood chemistry did the fixing automatically within its own bones without any human involvement, starting at the moment of death. Laboratory testing confirmed the effect directly. Hemoglobin exposure increased the stability of blood vessel tissue more than 200-fold, extending its survival at room temperature from roughly 3 days to more than 2 years. The same iron that had carried oxygen through the living animal's blood became, the instant that animal died, the very thing that sealed its tissue against the decay that should have erased it. Scale that same stabilizing effect across geological time inside a sealed, mineral-encased bone, and a route to 68 million years of preservation stops sounding impossible and starts sounding like straightforward chemistry. The final piece of this story addressed whether the 2005 discovery had simply been an extraordinary fluke, one uniquely lucky bone from one uniquely lucky animal that happened to preserve tissue no other specimen ever would again. In 2015, researchers examined several separate dinosaur fossil fragments that had sat in museum storage for more than 100 years, chosen specifically because they were unremarkable and poorly preserved, with no outward sign that anything unusual was inside. These were not carefully selected showpieces. They were leftover fragments nobody had thought worth examining closely before. Several of those pieces, dated to roughly 75 million years old, contained structures consistent with collagen fibers and red blood cells, complete with an internal nucleus, matching the structure of blood cells from living birds like the emu. None of these fossils had been treated with any special care during excavation or storage. They had simply been sitting in drawers. The implication was significant. This kind of molecular preservation may be far more common throughout the fossil record than paleontology had ever assumed, hiding in ordinary, previously studied bones that no one had thought to examine at this scale. None of this ever produced a genome. No blueprint exists, and none is coming. The chemistry that let collagen survive 68 million years does not extend to DNA, which remains bound by its own, far shorter, expiration date, no matter how favorable the conditions. Any hope of an actual living, cloned Tyrannosaurus rex remains exactly as far from reality after these discoveries as it was before them. What they produced instead was something science had never had before for a non-avian dinosaur. Real molecular evidence, not just from bone shape, but from an actual sequenced protein, tying Tyrannosaurus rex directly onto the same branch of the family tree as living birds. The proof did not survive because someone found a way to preserve it. It survived because the animal's own blood, in the final chemical reactions of its own death, accidentally embalmed the evidence that would confirm what it truly was, 68 million years before anyone existed to read it.